I apologize for my last video getting cut off, but to talk more about the fluorescent imagery that the researchers discovered, and also hyperplasia, which is where we left off, a hyperplastic outgrowth, which that basically means that there is a growth of extra cells that projects out of from tissues inside of your body. And hyperplasia is a sign of the early transformation or formation of cancer because it is defined as an increase in cell reproduction in the tissue or in an organ. And if this takes place in an organ, then you will be able to see this because it is enlarged. And that is a key sign to finding or to noticing whether you may have early cancer. So these are the images of the tumors that were generated, and this is a histological assessment of the tumors that were generated from the P53, P10, and RB1 combination, or the P53, P10, RB1/NF1 combination. And the histological assessment noted that that it reflected luminal B breast cancer, and this is kind of what I touched on earlier in the presentation. And luminal B breast cancer is classified as tumors which are positive for the estrogen receptor, and in this one you can see that ER in the third box up on the top. And one thing that was very promising with this experiment is that after researchers determined it was luminal B breast cancer, they were able to treat these tumors with drugs that target that estrogen receptor. To summarize this presentation in a nutshell, these findings suggested that breast organoids can be easily generated from breast tissue using the CRISPR-Cas9 technology, which modified human breast organoids, and there were donors, 12 donors. And also this CRISPR-Cas9, it showed that the editing of the tumor suppressor genes could be used to recapitulate ER plus, estrogen receptor plus, luminal breast cancer. One of their biggest findings was that in order for there to be achondrogenesis or tumor formation, there had to be a minimum of 3 tumor suppressor genes that were inactivated, or this could also mean that they were mutated. But this basically means that 3 tumor suppressor genes could not work in order for tumor formation to occur. And overall, this study was used to mainly understand the efficacy of CRISPR-Cas9 and the initiating events that give rise to breast cancer. And this is a topic that has not been investigated in great depth. So this research study really has promising findings, and we can really understand these initiating events that do predispose cancer and learn how to treat these cancers early. And there are so many different kinds of breast cancer. So understanding that these events, which are shared between the initiating events, are similar, even if they are different cancers under breast cancer. This was the research study that I did this presentation on, and I highly recommend it. It's very in-depth, and sometimes it was hard to understand, but once you really picked out the important parts of it and understood what their process was, what their methods were, how they analyzed this, and you can look at the beautiful images that they discovered. The fluorescent imagery was great to look at because you could really see that tumor formation. So I really highly recommend reading this.